Hello, and welcome to The Game Show! Today I have two new contestants who will be battling for a new Prius! Well, today's a very special day. Today we'll be focusing on the mysterious yet marvelous creatures known as Ctenophores. Right here is our first guest, Ike. Ike, tell us a little about yourself.
Hi, I'm Ike Girl, and I attend LMU. My favorite classes happens to be Biology 111. I'm so excited to be here.
Thank you, Ike. Okay, well, we have our next contestant, Becca Abarnes.
Hi, oh my god, I can't believe I'm here. Wait, really? Save the Ctenophores!
Wow! Thank you for the enthusiasm, Becca. Alright, guys, the rules are simple. I ask a question, and the first one that buzzes gets the answer. First question: how do Ctenophores move?
Okay, I got this one. So basically, they have 8 comb rows of cilia fused along the sides, right? And then these cilia, they beat synchronously and propel them through the water.
You are correct! Next question. What is the basic biology of Ctenophores?
I got this? Oh my god, wait!
Time's up! Alright! Ike, you may answer the question. Never mind. Alright, we'll let our trusty scientist, Brigitte Serna, answer this question for you.
Well, Sebastian, in reality, little is currently known about the basic biologies of most ctenophores. They are transparent, translucent, or bioluminescent, and range in size of millimeters up to 2 meters in length. They have biradial symmetry and an incomplete gut. They also lack an excretory system, a respiratory system, and a circulatory system. Most of these beautiful and exquisite organisms are rarely studied because they are either difficult to obtain or extremely delicate, or both.
Okay, ladies, next question. Where are Ctenophores found?
Hello. So, anyway, Ctenophores, they're marine animals, and they can be found in all seas and oceans, right? So they're native globally, but they can be invasive species locally. That's a good detail I threw in there just because I thought you might like it. And then, so they can be found from like coastal areas all the way to open waters, baby. Save the Ctenophores!
That is correct! Next question. What do Ctenophores feed on and how?
Huh? Oh. So, I learned this from my professors in my Bio 111 class at LMU. Okay, so, basically, alright, Ctenophores.
Do you ever look at someone and wonder what is going on inside their head?
Wow! According to the professors at my Bio 111 course at LMU, Ctenophores use sickly cells called coloblasts in order to capture prey. In a few species, special cilia in their mouth are used for biting glutinous prey. They consume most of the young of fish, larval crabs, clams, and oysters, as well as copepods and other platonic animals that would otherwise serve as food for such commercial fish as sardines and herrings.
Hold that. We now interrupt your daily scheduled program to present to you on the diversity of Ctenophore.
Have you ever wondered about the major groups of Ctenophora and their characteristics? Well, wait no more. We have the answers for you. Firstly, many Ctenophora can be found in a wide variety of shapes and sizes, from spherical to ribbon-like, and from less than a centimeter in diameter to over a meter in length in some ribbon-like forms. They are usually planktonic. There are just over 100 extant species of Ctenophora, which can be divided in just 2 classes, Class Nuda and Class Tentaculata. Class Nuda do not have tentacles, while Class Tentaculata do. Now, I will be asking the next question for our lovely contestants, Ika and Becca. How do Ctenophores reproduce?
I know this one. I know this one. Who's gonna call me? Nobody's gonna call me.
You can go, Becca.
Okay, thank you. Okay, so pretty much like they're hermaphroditic. So they have sperm and eggs, and they keep them in separate gonads, and then they like store them in the meridional canals. I never know how to pronounce that, but that's how we're pronouncing it today, so it's fine. And so they eject sperm and eggs through their mouth, and then fertilization and embryonic development occurs in the water.
That is correct! Okay, next question. What are the life histories of Ctenophores? Go ahead, go ahead.
I'm gonna hang this. Okay, well, I learned in my class, Bio 111, at the Bengal School of Science and Engineering at LMU, that forest development is indirect but not metamorphic. Fertilized eggs of most species rapidly grow into ciliated sediped larvae, which gradually obtain the adult sizes and morphologies. Individual species may have lifespans of anywhere from less than a month to 3 years. Moreover, while Ctenophores may have locally high population densities, they are solitary animals.
That is correct! Alright, next question, ladies. Do Ctenophores have any ecological impacts? And do they benefit humans?
I know this one. Okay, so no, they don't. No. Beyond scientific research and like public display, like aquariums, so cool. Save the Ctenophores!
That is correct! Alright guys, bonus round. Whoever can build a phylogenetic tree of Ctenophores gets extra points.
Okay, wait, but I can do this. What's phylogenetic?
Yeah, my Biol 111 class didn't teach me anything about that.
Okay. I guess we'll have our trusted scientist, Brigitte Serna, answer this one for you.
Okay.
For the 16S rRNA phylogeny tree, we see that ctenophores are closely related to Cnidaria, which they tend to be mistaken for. Then, they are related to Porifera. They are distantly related to Annelida. Thank you, Sebastian. As you can see on the phylogeny tree created from the data matrix in the Bio 111 class, ctenophores are closely related to Porifera and then Cnidaria, Porifera being the outgroup in this phylogeny tree. They are distantly related to Mollusca and Chordata. The trees may look similar to one another, but they are different. The trees differ with Ctenophora's closest relatives and their outgroups. Also, the relatives in general are in different orders. The tree created through DNA sequencing shows sister taxa between phylum, whereas the tree created with our data matrix only shows one pair of sister taxa, Mollusca and Chordata. These differences are due to the fact that one of the trees are based off of morphological traits, while the other one depends on DNA sequencing. These differences may be caused by the number of tissues, body cavity, digestive system, support system, and nervous system.
There is very little information on the origin of ctenophores. Since ctenophores' bodies are made up mostly of water, the chances of leaving a recognizable fossil are very slim. Until fairly recently, no fossil ctenophores were known. However, 2 species of fossil ctenophores have been found recently in southern Germany. But since these fossils differ from living ctenophores in several ways, this obscures their phylogenetic affinities. Historically, sponges were thought to be the sister group to the rest of the animals, a hypothesis that was consistent with the assumption that neural cell types arose after sponges and placozoa diverged from the rest of the animals. Recent findings from phylogenomics support the position of Ctenophora as a sister group to all other animals, challenging these long-held assumptions. Consequently, ctenophores are thought to be the oldest extant animal to possess a functional nervous system. However, there are still discrepancies found in different published phylogenetic trees. This phylogenetic arrangement opens the possibility that sponges and placozoans could have lost neural cell types. Thus, this phylogenetic tree supports the idea that neural cell types evolved once in the stem leading to the most recent common ancestor of all animals and were subsequently lost or modified in Porifera and Placozoa. While this is the phylogenetic tree arrangement that shows the possibility that the ctenophore's nervous system evolved independently from that of Cnidarians and the Bilateria. Thus, this phylogenetic tree supports the idea that neural cell types arose independently twice, once in the stem of ctenophores and once in the stem leading to Cnidaria and Bilateria. However, the phylogenetic position of ctenophores has been and still continues to be in dispute. Further analysis with better models, Fourier taxon sampling, or alternative markers will be needed to settle this question.
Thank you for joining us today on The Game Show!
Save the ctenophores!
Okay guys, the winner is... Drum roll please!